Hi everyone, my name is Mohit Kumar Jolly. I'm an associate professor here at the Department of Bioengineering at Indian Institute of Science, Bangalore. I run an interdisciplinary research group which has graduate students, postdocs, and undergrads from different backgrounds: ecology, computer science, mechanical engineering, physics, mathematics, biology, bioinformatics, all working on these questions of the two most deadliest aspects about cancer progression, which is metastasis and drug resistance. How cancer cells spread from one organ to another and how they develop resistance against different therapies that we have to throw at them. So currently there is very little understanding about the dynamics of these processes. So we use mathematical modeling as a tool to understand how these properties emerge as a consequence of interconnections between different nodes of molecules within a given cell, as well as the crosstalk between different cell types in a tumor microenvironment. So our goal here is to use this tool in close collaboration with experimental and clinical colleagues, which we do already here at IISc through joint PhD students and other collaborative mechanisms, and use this better understanding to then outsmart cancer at its own game of adaptation and survival, how cancer cells adapt themselves in real time through all these challenges, and then get better strategies in terms of therapeutic ideas.
Hi, I'm Vibhishan. I'm a postdoctoral research associate at Dr. Mohit Kumar Jolly's lab at IISc. I'm interested in developing new mathematical models for cancer that are inspired from ecology. Ecology is a discipline that tells us how animals and plants interact in the real world, and it turns out that a lot of the mathematics that we use to understand how that happens out in the wild can also be used to understand how cancers grow and cancers progress. So my interest lies in the interface of cancer and ecology, and how some of these ideas from ecology can help us understand cancer growth better.
Hi, I'm Soundarya and I'm a third year PhD student at the CSP lab. I think the most interesting thing about cancer cells is that it's constantly evolving and adapting to its new environment. One such example of this is when a tumor is growing, it has a stressful condition that's being formed wherein it doesn't get enough oxygen from the blood vessels. So that's a condition called hypoxia. So this is a problem, and this is a thing that to which cancer cells have to adapt to. And another interesting thing about cancer cells are that no two cancer cells are same and no two tumors are the same. Therefore, my goal and my plan for my PhD is to look at how the environment changes and how there is heterogeneity within the cells in a tumor. Thanks to technology, we now have a lot of single cell and spatial transcriptomics data where we can look at the gene expressions of an individual cell. This helps us understand how things change and how there is heterogeneity, and how to now understand the microenvironment and then proceed with strategies to then treat the tumor.
Hello, I am CVS Prasanna, a senior PhD student working with Mohit and Ramre in the Cancer Systems Biology group. I work with agent-based modeling approaches to simulate and study cancer metastasis. To give you an example of the kind of research I do, I would like to show you a small simulation. So I use a cellular perch model formalism and implement that in a tool called computational 3D, where I use it to study how cancer cells migrate from the primary tumor. As you could see here, a set of cancer cells which are originating at the center of the simulations are trying to invade into extracellular matrix, and they are trying to degrade the matrix and invade into it. So I induce a mutation in this tumor called epithelial mesenchymal transition mutation. So it is characterized by reduced cell-cell adhesion and increased cell ECM adhesion, which are typical mesenchymal traits, as you could see here. So the question which I was concerned about is, when does that mutation or the transition which has been induced thrives and dominates, and how does that influence overall migration in the primary cancer tumor? So as you could see here, the growth fields which kind of get generated from the transition cells affect the way in which the untransitioned ones move and the transition ones move. And sometimes and sometimes we see a phenomenon called confinement, in which the cancer cells, the transition cells get confined, and sometimes we see that confinement doesn't happen and the transition cells move. So in the recent work we kind of showed when which will happen, like when does the transition cells achieve domination and when it doesn't happen. Besides the adhesions and forces, I also work with other biophysical parameters like the cell stiffness, the sizes and shapes of the cells, and how does that influence the migration of the cancer cells.
Hi, I am Sajanya. I am second year PhD in Mohit Jolly's lab, and I have done my BSMS in mathematical science, and now I am doing my interdisciplinary PhD at IISc. My research focus on macrophage dynamics, where I see the gene regulatory network and how the network changes the behavior and the phenotype of the macrophage. The key direction of my project is to integrate those macrophages with the cancer dynamics and see how these two cells can help each other in the progression. Given a large diversity of students in our lab, it can help me to focus beyond a particular dimension and integrate a lot of discipline into my project.
Hi, I'm Ritesh. I'm a 5th year BSMS student in the lab, and what I work on is something called as phenotypic plasticity. What this means is cancer cells, which are very well known to be genetically identical, genetically clonal, may not exhibit the same phenotype. Let's say, for example, in terms of metabolism, a cell which is high in glycolysis and another cell which is again genetically identical to the previous cell might not be high in glycolysis. It may be high in OXPHOS, and you might have a third cell which is high in both glycolysis and OXPHOS, and all three of these cells are genetically identical again. And this kind of behavior is seen in multiple axes. It's been seen in terms of stemness, it's shown in terms of luminal and basal switching, specifically in breast cancer, and where this becomes important is in terms of drug resistance. What happens is a cancer cell which is to some degree responsive to a current drug, and another cancer cell which is again the same identical clone of the previous cell might not be responsive to the same drug to the same extent, in the same tumor microenvironment, in the same conditions, in the same concentrations also. And what my work primarily has been based on is showing that these axes of phenotypic plasticity, these multiple axes of phenotypic plasticity, are not independent. Let's say metabolism, let's say stemness, let's say drug resistance, let's say luminal basal are not independent, and these are in fact correlated. And not only they're correlated, but also they are interconnected in such a way that when you perturb one axis, when you push a cell from having a certain metabolic phenotype towards having a different metabolic phenotype, it alters these multiple different axes of phenotypic plasticity that we just talked about. Let's say drug resistance also get perturbed.